Hello everyone, my name is Xingfeng Shao from University of Southern California, and here I'm gonna present our work on vivo Laminar CBF fMRI using high-resolution pseudo-continuous arterial spin labeling and 7 Tesla. And I have nothing to declare. So arterial spin labeling is a non-invasive technique to measure cerebral blood flow. So ASL scan starts with a control session. You can see the flowing blood come from the feeding aorta and goes all the way to the capillary. And we repeat this scan again, but this time we invert the incoming blood and the feeding aorta with RF pulses. And after the same post-labeling delay, so those inverted spins arrives in the capillaries. So after a subtraction between the control and label images, here we can get a beautiful perfusion map. So one major limitation of ASL technique is the low SNR. The major reason is because ASL signal is only about 1% of the raw image intensity, and there is also T1 decay during the post-labeling delay. So lower SNR limits the capability of using ASL to detect the brain activity. So there are several benefits to do ASL and ultra-high field. So for example, the first thing is SNR increase superlinearly with field strength. According to this equation, you can see there should be about threefold SNR increase from 3 Tesla to 7 Tesla. And also specifically for ASL, the T1 of blood increase from 3T to 7T, and you can see a slower decay of your signal during the post-labeling delay, which means you can get a 30% SNR increase. So here's a theoretical simulation of the ASL signal as a function of the field strength. As you can see, there's a significant increase of the Picasso signal when you moving from 3 Tesla to 7 Tesla. And here's another previous study from our group showing you the perfusion images and different field strengths. So and ultra high field. So you can see not only you can get a higher in-plane spatial resolution, but also you can get a better image contrast and signal to noise ratio. So cerebral blood flow measured by arterial spin labeling is a key parameter for in vivo assessments of neurovascular function. Because the major reason is because the CBF measured by ASL is less affected by the susceptibility effects, for example in BOLD. The BOLD signal is mainly driven by the vein, but the ASL has higher specificity because it's closer to the set of neural activation in the capillaries. And the study also showed that the visualization of the orientation columns, the ASL has actually superior spatial resolution compared to both fMRI. Another animal study also showing that ASL CBF has faster hemodynamic response. The perfusion signal response arrives about 1 second earlier than that of the both signals. So if you read our title, I guess your first question is, does 70 really provide you sufficient SNR to measure perfusion or even activation in different layers, right? Because each layer has a thickness of about a few millimeters, so we definitely need a sub-millimeter resolution to separate the perfusion or activation in different layers. So in our last year presentation, we demonstrate that the layer-dependent CBF can be measured with a combination of the Picasso labeling, inner volume 3D acquisition, and optimized background suppression. Just each component has to be tuned super carefully. So here shows the experiment setup we used. The yellow box is a smaller FOV. We only interested in the motor cortex. So with a smaller FOV, we can have a much shorter echo time and a shorter scan time. So another benefit is we can move our labeling plane all the way from cerebellum to above circle villus. So with a closer labeling plane, we can have a shorter arterial transit time. And also we have the best B1 and B0 homogeneity in the center of the brain, so which is common a huge problem for scanning and 7 Tesla. So to measure activation, we also need to acquire enough measurements, which requires short TR and lower SAR. And 60 to 70% of SAR actually comes from the Picasso labeling pulses. So here's the simulated label and control signal with typical parameters we use for Picasso and 3 Tesla. And you can see the orange curve is inverted in the middle, which is a virtual labeling plane. So typically with this setup, it gives us about like greater than 90% labeling efficiency when we look at the ICA, which has the flow velocity about 40 centimeters per second. However, if we're targeting at MCA above the circle villus, the blood flow is actually 2/3 slower, with the same parameter, we can only get a 75% labeling efficiency, and not to mention the SAR might be too high. So we optimize the labeling parameters, so now targeting and the slower spins, so we can achieve 87% labeling efficiency, but with 40% reduction in the flip angle, which means 64% induction in SAR with optimized flip angle and gradients. And our MRI experiments were conducted on the 70 Siemens Terra scanner with single-channel transmission coil, and we get data from 4 healthy young subjects. So here are the imaging parameters for the Picasso. We had a in-plane resolution about 0.5 millimeter after interpolation and 1.5 millimeter through plane thickness. And we have optimized background suppression targeting for CSF. And our SAR was monitored under the first level, so provided by the Siemens scanners. For the fMRI tasks, we have 5 blocks of 60 seconds bilateral sequential finger tapping task, interspaced by another 60 seconds of rest. The TR of each scan is 10 seconds, and our total scan time is 10 minutes. So here in the first row, you can see the control image, and the second row shows the CBF maps we measured and resting state. So next click, so here you will see the CBF map we measured with finger tapping, right? So as you can see, the majority of the CBF remains identical, but if you pay attention to those areas indicated by the blue arrow, so we can see a strong CBF increase along the motor cortex. And you can also see some activation around the premotor area. So here we're zooming a small area around the motor cortex, and you can see a very strong CBF or the activation invoked by the finger tapping along the motor cortex. So for the next step, we did a surface expansion and within layer smoothing for better visualization. And we also did GLM to find voxels with significant CBF activation. So as we can see here, CBF activation can be reliably detected from all the 4 subjects we have scanned. So this is a plot of the profile of the average CBF. The x-axis follows the depth from the wet matter towards the CSF. The red curve is the CBF profile and resting state, and the blue curve is the CBF when we do the finger tapping task. So for the red curve, we can see in resting state, CBF has a peak in the middle layer. The green curve is the CBF activation invoked by the finger tapping. It's a difference between the blue and red curve. As you can see, the CBF activation also have one peak towards the CSF in the middle layer, and there seems to be another shoulder towards the wet matter. So in conclusion, the layer-dependent CBF activation can be measured using Picasso with inner volume excitation and 7 Tesla. For the baseline ASL, we found the highest blood flow in the middle layer, which is in corresponding to the highest capillary density in the cortical layer 4, as we can see here from the optical imaging results. And the ASL is also capable of detecting layer-dependent activation. The benefits of using ASL is because the CBF is quantitative and is less susceptible to the vascular contamination. However, it requires more technical developments to further increase the SNR. So in vivo imaging of layer-dependent CBF and ultra-high fields definitely open doors to investigating the neurovascular coupling in a laminar fashion. Alright, I'd like to thank my colleagues in USC and our Lonely resources, and also I would like to thank Beijing MR Center for Brain Research, Dr. Peng Zhang and Fan Hua, and also thank NIH for their funding support, and thanks Siemens for their technical support. Thanks for your attention.